Okay, part two of our lecture. We made a stop partly because I'm always a bit nervous with the Canvas challenges, but also because I think this was a nice segue. Did we cover? We covered really quite practical measures of markers of maturation. So, the ideal would be physician Tanner staging plus serially measured heights, weights, sitting heights, so we could get a longitudinal age at peak height velocity. But the practicalities of these things are we are reliant on self-assessment of Tanner staging and an estimate, a prediction of age at peak height velocity, often from a single measure. And there's a piece that's missing here that we haven't talked about. So our Tanner staging gives us a window into reproductive maturation because we're visually assessing the onset of secondary sexual characteristics. And secondary is the key word here. They are secondary to the release of hormones. Those hormones were released, and the release of those hormones has allowed the appearance of these sexual characteristics. But we haven't measured hormones. And why is that? That's a conundrum perhaps in the exercise sciences. We really haven't used growth hormone or any of these the gonadal function hormones, and there's a multitude of hormones that we're going to see that are really important in terms of reproductive maturation. So one of the reasons is simple. It's that the capture of hormones, particularly in females, and I'm thinking estrogen and progesterone, require blood. So you can actually record salivary testosterone quite well, but our ability to measure other hormones from saliva has been limited, and we're only just beginning to work on this now and improve those biomarkers. So let's talk a little bit about the hormonal control of growth. So, we've probably covered this, probably for you guys a long time ago in physiology. You probably looked at the maturation, the sexual maturation system, and the regulation of growth is complex. It depends on an interaction between a number of things, not least of which is hormones, nutritional state, the physical environment, and of course our genotype, and the potential that genotype provides for the individual. But the hormones are of importance here, and they're important because of the initiation and regulation of pubertal events, so things like the appearance of secondary sexual characteristics, attainment of adult reproductive function, peak height velocity, all of these are controlled by hormones. So some of the most prominent hormones involved in the regulation of growth and maturation include testosterone, estrogen, thyroxine from the thyroid gland, cortisol and adrenal androgens, insulin, and a series of hormones from the pituitary glands. So we can consider growth hormone as our overview, and it stimulates the liver to produce insulin-like growth factor 1. And at puberty, growth hormone interacts with the gonadal steroids, the testes in the males, the ovaries in the females, to release some of those other hormones, estrogen, testosterone, and so on, to complete our progress toward the adult state. This central control of growth and development is comprised really of a three-part system known as the hypothalamic pituitary gonadal axes. So you can see here in our little picture on the right, the hypothalamus in the brain, the pituitary gland, the thyroid, we get the adrenal glands, and then of course the gonadal ovaries, testes, those involved in the androgens and the gonadal hormones. So our hypothalamic pituitary gonadal axes, our HPG, everything else is downstream to this, and that's the central feature of how the signaling for growth starts, and specifically maturation. So we'll talk about this in a moment, about how the central features start to flow between the hypothalamus, the pituitary gland, and we have the release of a variety of hormones. But probing question for you is, does exercise impact or affect the growth hormone and insulin-like growth factor 1? So taking that into the context of our example with gymnastics, this is an older study now from Dan Nemeth and his lab, and it was published back in Pediatric Exercise in 2013. It was a really neat study that looked at the impact of training of different amounts on two factors. One, insulin-like growth factor 1, and the other, a biomarker of inflammation, IL-6 or interleukin 6. So if you take a look at this graph, you can see on the top graph you've got insulin-like growth factor 1, and you've got the bottom graph, you've got interleukin 6, our biomarker for inflammation. And at the bottom here, the x-axis, we have a pre-season period, light training, very heavy training, then a tapering down of training to leading up to competition. And what's super interesting that they found was that IGF-1 declines with very heavy training, but when the youngster tapers, it bounces back. So it's back up and above baseline levels for competition. So this is a really interesting dual play. This is stress on the system. If you carried on training that youngster, very heavily over time, you would maintain that reduced IGF-1. And when there is stress in the system, and the stressor here is the heavy training, then you were going to get inflammation in the system. And this was picked up nicely with the biomarker for inflammation, IL-6. So with that heavy training, there's now a surge in IL-6, and it's chronically elevated. If that youngster stays overtraining, it will remain chronically elevated. And that's been associated with stunted growth. So this might be an adaptive mechanism. So IGF-1 may reduce because there's got to be a reduction in this kind of bodily global anabolic function to conserve energy. It will still allow for some tissue growth, but this is in response to this big stressor like heavy exercise training. And consistent with this speculation is this idea that you will get attenuated somatic growth with this reduced circulating IGF-1 with intense training, specifically in nutritionally deprived young female gymnasts. So there is some data out there that says this can be a problem, and it can be a problem that's specifically driven by the stress of too much exercise, heavy, intense training that persists over time. But the point here is also that it is in concert with nutritional deprivation. So let's just go back to our central features of our HBG loop, our hypothalamus pituitary gonadal system. Top down, remember, all starts at the hypothalamus. And the hormone signals flows from the top down component. The hypothalamus serves as the main signaling, and it signals a hormone called gonadotrophin releasing hormone, GnRH, or luteinizing hormone releasing hormone, which is LHRH. And these are released in a pulsatile fashion. And these are an important part of the on-off switch, if you like, for controlling reproductive function. So the effect of GnRH release is to stimulate the production of the two gonadotrophins, the FSH and the LH. In the ovary, LH stimulates testosterone and FSH stimulates the conversion of testosterone to estradiol. On average, LH concentrations rise first in girls at about 8 to 9 years of age. That's reflecting their earlier onset of puberty. And then in boys about 2 years later. And at the same time, LH is produced, inhibin is secreted and has a suppressive effect on FSH, and progesterone then is secreted. And this combined effect of these steroids and the inhibin is to inhibit the pituitary. And in the testes, LH stimulates testosterone production, and FSH stimulates inhibin secretion and acts as a feedback mechanism on that pituitary gland. So gonadal function is regulated by other metabolites such as insulin, and we see that with our insulin-like growth factor, and of course cortisol and growth hormone, which we've just seen in the example study. So elevations of any of these gonadotrophins are associated with maturation, evidence in testicular volume in the boys, and the appearance of those pubertal changes that we see: pubic hair, facial hair, voice changes, accelerated growth, increase in muscle mass. Characteristics in the females would be the same, with breast enlargement, pubic hair, axillary hair developing, and so on. And then it also starts to increase linear growth. And then there are some changes in dimensions, so the pelvis remodels. There's an increase in the girls specifically in adiposity, and of course the onset of menarche. So if we look at testosterone and estradiol levels during puberty, this is from a textbook that gives you a nice visual of this. The boys in testosterone in the left-hand graphs, the girls for testosterone in the right hand, and then estradiol in the lower right, left and right hand for the boys and girls respectively. And these elevations in the gonadotrophins are associated, as I said, with that gonadal maturation, and you see a resultant rising testosterone. And these are our Tanner pubertal stages, and these are now plotted against our hormones. And so you can see this very, very steep rise in testosterone in the boys across Tanner 1 to 5. And in the girls, there is an increase, a bit of a plateau, and then a secondary increase just at the end of puberty in Tanner stage 5. If we look at estradiol, of course, that estradiol is secreted in boys, but to very minimal amounts. But you can see a parallel to the testosterone in boys, there's hugely steep incline in estradiol in the girls across the pubertal stages. So our hormone control of puberty is lining up to a degree with our Tanner staging. And if we look at this one specifically for girls, we can see where menarche falls, and we can line up all of these different facets from the hormonal to our height and body mass velocities, and where each of these fall. And you can see here that this is our growth hormone. It peaks just after height. So this is really that linear growth in stature, but also that growth in body mass. We can see that the IGF-1 is peaking somewhere around Tanner 4. And we can see the estrogen rising, onset of menarche. So both IGF and specifically estrogen have risen sufficiently for the onset of menarche, but estrogen will carry on to rise till it attains the adult value. And there is a rise in testosterone in the girls, but it plateaus from about Tanner 4 onwards. So you can see that the onset of these hormones have occurred. Growth hormone has already started to be released in Tanner stage 1. There is an increase between Tanner stage 1 and Tanner stage 2, somewhere in that age range of testosterone, but specifically of estrogen and testosterone, and of course our growth hormone. But both growth hormone and insulin-like growth factor start to decline after the growth spurt, and you can see this very dramatically once we've attained our somatic size. So that's just a little piece around the hormonal measures. It's important that we think of them because Tanner staging does not happen without the gonadotrophin hormones. Peak height velocity does not happen without growth hormone. And also the gonadotrophin hormones. We start to see these changes in body composition, and this is all very important. So just for our next class and next week, we're going to consider sport, and specifically we're going to consider the debate around the importance of energy availability in the young athlete and how this very heavy training may interface with stunted growth or not. So there's a couple of things that will come up on Canvas. One is a neat paper just looking at the selection bias in sport, and I think that's really important as a pediatric exercise scientist for you to consider when we don't take into consideration maturation, and very importantly for our case study, this issue around energy availability and how that's interfacing with our young athlete. I will see you on Wednesday.